Bismillah ar-Rahman ar-Rahim. Our lecture today about the gene structure and the chromosomes. Actually, this is the basis of the human genetics. Here to this slide, you find that the violet in the first generation is R capital, but the violet in the first generation is R capital, R small, and the violet in the second generation may be R capital, 1 and 4, and R capital, R small, R capital and small, 2 and 4. So we have two phenotypes, white and violet, but we have three genotypes, R capital, R small, and R capital, R small. He said that there is dominant and recessive character. So the violet is dominating the white. So the R capital is for violet and R small for the white, and the white doesn't appear except in the homozygous state. The third law is the law of independent assortment if we are talking about the color and the size of the seeds. So the color has two genes and the size of the seeds has two genes. If I start by violet and large seeds and small and white seeds, in this case the size and the color is not associated together. I mean that in the first generation I may have large violet, large white, small violet, small white. So there is interchange of the character. So not essentially all the traits are running together, except if the genes of the two traits are very close to each other in the chromosome, and hence he said that the traits are linked together. The laws of Mendel, the three laws of Mendel are the fundamentals of the science of genetics, but sometimes they are violated or breached, and this is the progress in the branch of human. The number of the chromosomes in the human cell are 46. They are present in pairs. So there are 23 pairs of chromosomes. So the human cell is called diploid cell. The appearance of the chromosome, as in this slide, in thread-like structure occurs only during the cell division. However, in the resting cell, the chromosomes are entangled together in the form of histone. So to have the chromosome in this appearance, I have to induce the cell to divide. So the human male cell contains 44 autosomes and XY, 6 chromosomes. However, for the female, it is 44XX. And each chromosome has two homologous pairs. One is coming from the father and the other is coming from the mother. During the cell division, the somatic cell divides by meiotic division to form the haploid germ cells, or the half number of chromosomes. It's present in the germ cell whether it is in the sperm or ovum. So the sperm is 22 autosomes with X or Y, but the ovum is 22 autosomes with only X. See, for the nucleus of the diploid cell, the chromosomes are present in pairs. The biggest chromosome is chromosome number 1, and the shortest chromosome is chromosome number 21 and 22. So this cell, during the meiotic division, during spermatogenesis, it divides into two sperms. Each one contains 23. This is one with X and this is sperm with Y. You notice that the chromosomes are present in the head of the sperm because the head contains almost all the nucleus, but the midpiece of the sperm contains the mitochondria with its mitochondrial DNA. Oogenesis results in the formation of only one type of ovum. All of them contains X chromosomes. So the type of the sperm is the one dividing the type of the sex of the offspring. During fertilization, sperm penetrate by the head only into the ovum to form the zygote, and rendering the number of the chromosomes again 46. Cell division is of two types. The first type of cell division occurring in gonads is called meiotic division, and this is either spermatogenesis in male or oogenesis in female. However, the mitotic division occurring in all over the body, and this is responsible for the growth of the organism. Actually, cell division under tight regulation to repair any abnormality or any aberration in the DNA replication, to correct any mistakes, and this is usually occur through checkpoints or restriction points. And these checkpoints, if it's minor, it can be repaired. However, it's major, so the cell will undergo programmed cell death, which is called apoptosis. Here in this animation, you will notice that during the cell division, the incomplete division of the cell into two incomplete disjunction of the homologous chromosome will result in what's called aneuploidy or trisomy 21. In the panel on the right side, you will see the non-disjunction. This means that the cell is divided not equal halves, but in the normal cell division, the cell is divided into two equal halves. Look and follow the movements of the animation. The homologous chromosomes are arranged along the equatorial plane. Here you notice that the cell is missing one short chromosome, but the cell contains two copies of short chromosome. But here the cell contains two equal halves. Continue again. Another cell division. When each of these cells starts to be fertilized by a sperm with half chromosomes, this will result in the cell on the right side contains three copies of chromosome 21, so it results in trisomy 21. This is called non-disjunction or incomplete disjunction, but here there is complete disjunction of the homologous chromosome, and this is responsible for the phenomena which is called aneuploidy. The chromosome in its appearance is composed of short arm, which is assigned by the letter P, and the long arm is assigned by the letter Q, and there is a constriction in the center which is called the centromere. And according to the position of the centromere and the size of the chromosome, we have classification of the 23 chromosomes into 7 groups. These are types of chromosome according to the position of the centromere. If the centromere is near the center, it's called metacentric chromosome. If it is shifted little bit distal, it's called submetacentric chromosome. However, if the centromere is at the end of the chromosome, it's called acrocentric. In acrocentric, there is no short arm, as you see. It contains a satellite. This satellite is different from the short arm is that it doesn't contain DNA or genes. So if the satellites are lost, the subject will not suffer from any DNA loss or DNA gain, but it will result in the phenomena which is called the Robertsonian translocation, which is one of the types of the Down syndrome. The arrangement of the chromosomes into 7 groups, as you see, group A, group B, C, D, E, F, and G. The chromosome number 1, 2, 3 are in the group A, 4 and 5 group B, group C from 6 to 12, and group D from 13 to 14 to 15, and group E 16, 17, 18, group F 20 and 19, and finally group G 21 and 22. The chromosome number X belong to the group C, and the chromosome Y belong to the group G. Here, which is containing the 21, 22, and Y chromosome. Notice here the size of the chromosome is decreasing from chromosome number 1 up to chromosome number 21 and 22. You notice that the chromosomes of each group, 1, 2, 3 of group A, are different. All of them are large metacentric, but they differ from each other by the distribution of these dark and white bands. So actually we have two factors dividing the 23 chromosomes into 7 groups, which is the size of the chromosome and the position of centromere. And the third factor which differentiates the chromosome in each group is the banding technique, or the banding of the bands and distribution of the bands on each. This is the chromosome number 1, completely different from the bands of chromosome number 2, and completely different from the bands of chromosome number 3. So these 3 chromosomes are one group. 4 and 5, another group, different. 6 to 12 are the third group, and also it is bands. This is the first acrocentric chromosome, 13, another acrocentric chromosome, and and a third acrosome. You notice here the P for the short arm is replaced by hatched part which contains no genes. So there is no banding in these satellites. 21 to R. Now we have studied the structure of the chromosomes and the groups and differentiation of the individual chromosome from each other. So we will go to the molecular genetics, or the study of the genes or the basic units that form these chromosomes. Actually, the first discovery of the DNA structure of the double helix DNA was on 1953 by Watson and Crick, and they were awarded the Nobel Prize independently by halves for their discovery. They discovered that the DNA is composed of double helix. The DNA is composed of basic unit which is called the nucleotides. And this nucleotides is composed of three structures: the sugar, phosphate, and nitrogenous base. And this nitrogenous base, as you know, either purine, which may be adenine and guanine, and pyrimidine, which includes cytosine and thymine. However, uracil is present in the RNA. The RNA means ribonucleic acid, which is present in the messenger RNA, ribosomal RNA, and transfer RNA. However, the DNA, which shows the structure of the gene, is composed of desoxyribose. It is double helix, and it contains thymine instead of uracil, and it's double strand. So RNA is single strand with ribose, and DNA is double strand with desoxyribose. RNA contains thymine, but the DNA contains thymine, but the RNA contains uracil. If we look for the basic structure of the DNA, you will notice that the structure of DNA is like the ladder. The sides of the ladder, as you see, is composed of sugar and phosphate, sugar and phosphate. And the steps of the ladder is composed of the nitrogenous bases. It's either in the form of adenine with thymine with a double hydrogenous bond, or guanine with cytosine with triple hydrogenous bond. So if I give you the sequence of bases in one strand of the ladder, I say that A, G, T, A, C, G, you can predict the sequence of bases on the antisense strand. It will be T, because A is always attached by T and G always attached by C. So you predict the other base by knowing the first one. So it is complementary binding between the purines and pyrimidines. Okay. The strand has one upward end or upstream. It's called 5' for the phosphate, and has a downstream end which is called 3' end. Of course, the double strand is not one lamin or one laminar layer, but it is double helix and twisted together like this picture. Here you notice that adenine is always attached with thymine, and guanine is always attached with cytosine. So they are complementary to each other: adenine with thymine with double bond, and guanine with cytosine with triple hydrogenous bond. Here in the structure of the DNA, double strand starts how the DNA strand is assembled or is positioned inside the nucleus in the form of chromosomes. Look here, the double strand is twisted around a protein, which is the yellow one, to form what's called nucleosomes. And these nucleosomes are packed together to form what is called the chromatin thread. And this chromatin thread is twisted again many times to form the chromatin mesh. And this chromatin mesh is compact together to form the chromosome. In the next animation, you will see how this double strand DNA is assembled in three-dimensional compact and condensation many times and coiled together to form and to be assembled finally in the form of chromosome. Look for this animation.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. They form around the time cells divide when the two copies of the cell's DNA need to be separated.
We noticed in the previous animation how the homologous chromosomes are detached from each other during the cell division. So to see the chromosome in a thread-like structure, we have to induce the cell into cell division, and which is called karyotyping or cytogenetic analysis. Now we are going to the DNA replication. In order the DNA to be doubled, arranging the cell for cell division, it must be replicated. So the double-strand DNA is splitted into two separate strands, and each strand represents a template which make a complementary strand. So the net result, we start one double strand and we end in two similar double strand to the original one, and this is called replication. I mean that the DNA is duplicated, arranging the chromosomes for duplication to form two cells, two daughter cells similar to the mother cell. After knowing the DNA replication arranged for cell division, we have to say how the gene is controlling the trait. The gene is controlling the trait by what's called the gene expression. Actually, the gene expression is called the central dogma. Central dogma is composed of two main processes. The first process is called transcription, in which the DNA is changed into a message, which is called messenger RNA. This process is called transcription. Transcription results in the formation of messenger RNA, which leaves the nucleus and go through the pores of the nuclear membrane to the cytoplasm, arranging the process for translation that it occurs at the ribosome to form the gene or the messenger RNA to be translated into a protein. And this protein may be a functioning protein like enzyme or hormone, or a structural protein like the cell protein, cell wall protein, receptor protein, etc. So the gene is controlling the trait by forming a journey from the messenger RNA to the protein structure. This is how the genetic code from the DNA is translated to the RNA and finally to protein. We have in nature about 20 different amino acids, which constitutes all proteins present in nature, whether for human or animal or plants. We have 4 bases, and each 3 successive bases are called the codon, and this codon is a code for amino acid. So we can predict from this 4 bases, 4 to the power 3, about 64 codes. So we have to have 46 amino acids, and actually we don't have except only 20. So how we can solve this? If you look for this table, you can say the different codes of these 4 bases. For the first letter, it may be 1 of 4, and second letter 1 of 4, and the third letter 1 of 4. So we have about 64 codes. We have 64 codons, but actually we have some codes for one amino acid. We have 2 for the phenylalanine, we have 2 for leucine, and 4 in addition to 4 to this. So leucine has 6 codes: 1, 2, 3, 4, 5, 6. So not each codon is a symbol for certain amino acid. We have for one amino acid more one codon to be translated from. Also, we have 3 conserved codons, which are called stop codons. It's called TAA, TAG and TGA. This is called stop codon. So we can explain the huge number of codons and the limited number of amino acid by the repetition of the codes for certain amino acid and also for the 3 stop codons. Here you can notice that here we have the nucleus. This is a green circle, and this is the nuclear pore, and this is the cytoplasm. First, this part of the chromosome, this is the chromosome, and this part of the chromosome is called gene. After zoom in, it's composed of segments, blue segments and yellow. The blue segments are called exons, and the intron is the yellow. And after transcription is changed into the messenger RNA by removing the introns, and this is called splicing out. So the remaining exons are united together to form the messenger RNA. It leaves the nucleus to the cytoplasm arranged for the process of translation. We have the upstream end, or the 5' end of the gene, which is the starting point, and we have the downstream point of the gene, which is the end of the gene. Here at the cytoplasm, the ribosome starts to be fixed to the upstream of the gene and read the first codon. It is GGC. So it calls for the transfer RNA, which go and pick up the corresponding amino acid for this code, which is arginine, and put it and the second codon for the leucine, and etc. So a sequence of codons will give a sequence of amino acids or a polypeptide protein, and this polypeptide protein is the one which controls the genetic trait. And now we go to the basic structure of the gene. Any gene is composed of 3 main components. If you look for this chromosome, if I unfold the chromosome to its constituents, double strand, double helix DNA, any stretch of the DNA between 2 points that control certain protein is called the gene. So the gene is any functioning unit, part of the double strand DNA of the chromosome that control the formation of certain protein. It's called chromosome. It's called a gene. The basic structure of the gene is 3 main parts. The first part is the coding sequence, which is called the exon, and the exon is represented by these blocks. So this gene is composed of 1, 2, 3, 4 exons. The second part is called the introns, and the intron is non-coding, and it is the parts present between the exons. So we have 4 exons, but we have 3 introns. And the last part of the gene is the regulatory sequence. It is the part at the start or the upstream, which is called initiator or primer or priming codon. It starts at the upstream at the 5' end, and the downstream part of the gene is called the terminating or polyadenylation end of the gene. Here we have certain terms which we have to mention in order not to be interchanged while talking about genetic terminology. We have the homologous chromosomes, the chromosomes which are identical to each other. We have the homologous chromosome number 1, homologous chromosome number 2, etc. So we have the locus. The locus is a part of the chromosome in which the gene is present. But the locus contains the gene, and the gene on both homologous chromosomes not essentially to be the same, may be different, may have one allele here and one allele there. So the alleles are the type of the genes in the homologous chromosomes, but the gene locus is the part of the chromosome in which the gene is present. We have, if the 2 alleles are identical, I say that it is a homozygous state. If the 2 alleles are different, I call it heterozygous state. The heterozygous states not essentially to be a mutant. I may have the 2 alleles different, but they are polymorphic. I mean that the allele here is normal and the allele there is normal, but both of them are not identical. So the term homozygous means the 2 alleles are identical, but the heterozygous means that the two alleles are different. Maybe one normal and the other mutant, and we have here mutant and mutant, but they are different from each other. How can I localize the gene on the chromosomes? Actually, we have two methods. The first method, which is crude, it's called the cytogenetic method, which is used for localization of the gene on the chromosome under microscope. So it is a visual method. And the second method, which is accurate and very precise, it is used for localization of the gene on the chromosome based on the number of the bases. Going first to the cytogenetic localization, here is the chromosome by its components. Is the short arm, long arm of chromosome number 7, and here is the centromere. Suppose I want to localize the gene name as CFTR. This CFTR is the gene for cystic fibrosis. This gene is present in this area. How can I label this area in this photo? I say that the gene is present here. This part is called subband 2, which belong to the band number 1. And band number 1 belong to the region number 3. And region number 3 is part of the long arm of chromosome number 7. So I put the label or the address of this gene. It is present in 7q31.2. So it's present in the region 3, present on the long arm of chromosome 7, that in the band 1 and subband 2. Actually, this previous method is crude and rough and not accurate. Why? Because in this area there may be multiple genes. So how I can differentiate between the multiple genes in one subband? So I want a more precise method. This is called the molecular method. It's based on the basis, and this is accomplished by the finishing of the Human Genome Project at 2003. If I call that the ApoE gene is present on chromosome 19, it starts from the 50,100,801 base pair, and it ends by 50,104,488 bases. This means that if I subtract this number from this number, I have the size of the gene. The difference between these two numbers represents 3,588 base pair, and this is the actual size of the gene itself. So the distance from the start to the end is the size of the gene. So this method is very accurate because it is based on the basis of each gene on the chromosomes. Actually, the human genes are not evenly distributed in the chromosomes. We have about 30,000 genes, and these 30,000 genes represent the human genome, the nuclear human genome, which is 3 multiplied by 10 to the power of 9 base pair. This billions of bases constitutes the 30,000 genes. These genes are not evenly distributed, but they are present sometimes heavy distribution in certain areas of the chromosome, and there are some chromosomes which are very rich in genes and the others are poor. The centromeric region, the region which is in the center of the chromosome, actually it's very busy during the cell division. Therefore, it's mostly non-coding. That's to say, there is no genes in the centromeric region. Actually, the subtelomeric region, which is the area of the locus present at the end of the chromosome, is the one which is very rich in genes. Therefore, any subtelomeric deletion usually results in severe clinical manifestation and even lead to severe mental retardation. Chromosomes with high concentration of genes are 19 and 22, although they are short in size. But chromosome like 4, which is very big, and chromosome 18 is relatively poor in genes. So the number of genes of chromosomes are not dependent on its size. Number 2, the number of genes on the chromosome are usually not in the center, but at the end of the chromosome. This is an example of the gene, and this is the exon 1, 2, 3, 4, 5. So this gene is composed of 5 exons with 4 introns. During the formation of the RNA, the introns are spliced out and removed, and the remainder messenger RNA. This is the pure mature messenger RNA constituting only 5 exons. An example for gene structure to OTC. This is a gene for a protein which is called enzyme ornithine transcarbamylase enzyme, which is present in the urea cycle of the liver. This gene is composed of these exons. Exon 1 composed of 669 base, and exon 2 139, etc. We have 10 exons, and these 10 exons constitutes this statistics. Here, look for the statistics of the gene of OTC. You notice that the coding basis in the 10 exons, we have some non-coding intragenic DNA, and this coding of the bases include 1062 bases, which constitutes, if you divide this number by 354 amino acids. So the protein of OTC is composed of 354 amino acids, and the whole structure of the gene, including the non-coding introns and non-coding parts of the exons, include 1921. If you look for the protein structure, you notice here that the 354 amino acid is substituted in certain point. Instead of being R, it will change it to Q. So the arginine is replaced by glutamine in the position number 109. And so you can translate the change in the DNA in the change in the amino acid, and hence it will be affected the enzyme activity. Thank you.